Here we go. Hello, everybody. I'm Matt Taylor. I'm in the Department of Microbiology and Immunology. I'm a virologist. I used to study herpes viruses until last year. I jumped into uh, driving some of our live virus research, looking at SARS coronavirus infection in cells. So the pandemic has been going on for over a year now, first starting a little before Christmas of 2019 in Wuhan, China, and rapidly spreading across all 7 continents. Antarctica succumbed to COVID infection just before Christmas Eve of 2020. There have been 111 million people infected worldwide, almost 2.5 million deaths. Domestically, we have hit a landmark of having over 500,000 people in the United States succumb to COVID infection. Case fatality rate has varied from between 0.03 to 0.5%. I don't think I really need to stress the fact that this is a very serious problem that has changed the way we all approach our life. And it's all caused by this little bug here. It's a virus known as a coronavirus, SARS coronavirus number 2. These are so small we can't see them by the naked eye or with a microscope. We have to go down to electrons to visualize these viral particles that transmit from person to person, causing this disease. On the right is just an image of what that electron micrograph looks like. It's a spherical virion. This is that, again, that infectious particle surrounded by this crown of, of what's been figured out to be the protein. This is the spike protein. And the spike protein is going to come up a number of times because it's incredibly important, not only for how this virus transmits and infects cells, but also how we have developed our strategy to control it with vaccines. This is a schematic of that virion on the right. It is an RNA virus. Its genome is packed inside, tightly wound with this nucleocapsid protein in a helical symmetry. It's wrapped in a lipid envelope, which means it's really susceptible to soap. So if you take away one thing, stop touching your face and wash your hands, because it's a really effective way to combat these envelope viruses. But I also want you to pay attention to these orange spike proteins. These are the proteins that mediate entry of the virus into the cell. They're also the target that is being used in that vaccine strategy. Immune responses to this spike are amazingly effective at blocking infection. Now, SARS coronavirus causes this umbrella of diseases we've termed COVID-19. It gets its name, severe acute respiratory syndrome virus, for the significant lung pathology. There's a lot of inflammatory infiltrate, cytokines, and immune responses that compromise lung function, causing people to have trouble breathing and sometimes a lethal illness. But that's not to say it doesn't infect other tissues. In fact, we found SARS in the brain, the kidneys, the liver, the intestines, and the heart. And all these tissues can suffer from damage of SARS coronavirus infection, resulting in a very wide range of illnesses. Some that are acute and self-resolving, some that last for extraordinarily long periods of time, where these people cannot seem to clear either the infection or the associated illness, this so-called long COVID. What we have learned, though, from people that have survived from COVID infection is the kinds of immune responses that are protective. And one of the critical features, not the only feature, but one of the major ones that is focused on, is the production of antibodies that recognize the spike protein. So as I mentioned, that virion, when it needs to enter the cell, needs to bind to a cellular receptor known as ACE2, at which point the spike facilitates that entry of the virion, initiation of infection. Antibodies that bind to the spike prevent that activity from occurring, neutralizing the virion and interfering with the process of infection. We've used this for some therapies, such as the Regeneron and other monoclonal antibody cocktails that have been given to severely ill individuals, but it's also the goal of most vaccines to derive an immune response that is going to protect against infection. Besides that, this pandemic's been going on for a year, the rate of vaccine development has been incredible and unprecedented. As early as 2 months into the pandemic, we already had vaccines in development that have already now received approval, faster than any other approval that has been seen to date for an infectious disease, but only because they built upon technologies that have been in development for decades. The leading candidates are part of these 4 approved vaccines, which comprise 2 different strategies: a nucleic acid or mRNA vaccine, this is the Pfizer and Moderna, as well as these recombinant viral vectors that use replication incompetent adenoviruses. That is not to say that all vaccines have been successful. We've had a couple that have been abandoned, but that is not to mean that they were flawed. It just means they weren't as good as some of the other vaccines that were in development. Just because this is fast, just because it is happening at an unprecedented scale, does not mean that these vaccines aren't any less effective. In terms of the strategy for these vaccines, they both are working on the same principle, which is to deliver a nucleic acid that will result in the expression of that spike protein by your cells to be recognized and cleared by your immune system. The Pfizer and Moderna vaccines are these mRNA expression vectors. They are RNA that's packaged inside a liposome, sort of think of it as like a balloon that gains access to the cell, delivering this message that is going to get translated to make the proteins. So your own cells are making these proteins. There's no risk of an infectious virus being produced from an mRNA mechanism. It's an extraordinarily safe system that has been, again, developed for a long period of time based on a large amount of very sound research. It has resulted in a highly efficacious response. Over 90% of people are protected from hospitalization or death from COVID-19. They elicit strong antibody responses. The limitation is that they require extraordinarily cold storage conditions. Now, the other major strategy that has been receiving recent approval are these adenovirus-based vectors. This is the Oxford AstraZeneca vaccine, the Sputnik vaccine out of Russia, and the new Johnson & Johnson vaccine. These are non-replicating viruses that do the same thing. They deliver a nucleic acid to result in the expression of that spike protein in your own cells to be presented and cleared by the immune system. These are also very efficacious vaccines. They are reducing hospitalizations and they're preventing infection. They are also having the benefit that Johnson & Johnson vaccine is showing great efficacy towards a single dose delivery. Not only that, but they are simpler to store and they have better stability than these mRNA vectors. And so it comes to this contrast of the benefits of different strategies towards creating a most efficacious vaccine, which is a measure of how effective that vaccine is. We have to consider it from multiple levels. Is it safe? Is it effective? And is it practical? Safe means that it has to have minimal side effects. It cannot be worse than the disease we're trying to prevent. In fact, all of these vaccines have shown minimal side effects, though people are worried. I will tell you from personal experience, it is no worse than a tetanus shot. If you've ever had a tetanus shot, you know your arm's sore, but it's better than getting tetanus. They're also very effective. They elicit good, strong, protective responses that prevent illness. We can measure that, like this study here from the Moderna vaccine that compared a group of placebo versus people that were given that mRNA vaccine. These are people who have been diagnosed with COVID-like illness or COVID illness, and showing that these people that have received the vaccine are at a significantly reduced risk of developing any amount of disease. That's where your 95% efficacy comes from. We also have to consider that practicality. Are they stable? Are they easy to administer? And they're not cost prohibitive. All of these measures are going into that idea that we are trying to convince the public that there is a greater benefit to receiving the vaccine than the risk to getting the illness in the first place. But vaccines are just but one part of our public health policy to prevent the spread of infection. This is a great model that came out about 6 months ago. It's known as the Swiss cheese model, which considers that there's no perfect prevention to get to blocking the transmission of COVID in our communities. But by using a series of good, not great, but good measures, we can significantly impair the spread of the virus from person to person. This includes the use of masks, staying home when you're sick, washing your hands, not touching your face, and vaccines are that last measure. We can control the spread in our communities if we use all of these judiciously.